The broadcast is now starting. All attendees are in listen-only mode.
Hi, good afternoon, everyone. This is George Orling from HTSA, and welcome back to the research webinar series after a couple-month hiatus for summer break. Hope everyone had a great summer, and we have a great way to kick it off for the fall with Chris Kay from the University of British Columbia, who will be presenting to us on the question of, is HD more prevalent than they thought? But before I get started, or if Chris gets started, I just want to make sure everyone who may be new to the webinars or the GoToWebinar software, how to ask questions, you can ask at any point during Chris's presentation. Just click in the questions box. It's highlighted in red. Type your question, and at the end of Chris's presentation, I'll ask these questions, as many as we can, on your behalf. I'll also point out that we are recording this webinar, and within 1 week after this, we should have this on our website as well as on our YouTube channel, which can be found on our htsa.org website by clicking on the YouTube icon highlighted here in the red circle where it says Connect with Us. So as I mentioned, I'm very excited to have Chris Kay here with us. Chris holds a bachelor's degree in molecular biology and philosophy from the University of West Florida. He actually immigrated to Canada and started working as a technician in Dr. Michael Hayden's HD research laboratory at the University of British Columbia before he began as a graduate student, where he currently is a PhD candidate in Michael's lab at the University of British Columbia. His doctoral dissertation is describing the population genetics of Huntington's disease mutations, and he plans to graduate in May of next year. So Chris's talk is, I've invited Chris to present on hopefully a paper that I think many of you have seen in the news or press releases lately. It was entitled Huntington's Disease Reduced Penetrance Alleles Occur at High Frequency in the General Population. It's also a topic of a recent HD Buzz article. So I know there's maybe a lot of questions about this exciting data that Chris and Dr. Hayden have identified. So I'm going to turn it over to Chris to walk everyone through it. Make you the presenter, Chris.
Yep.
And the floor is yours.
Okay, so can you see everything?
Yes, I can.
Great. Hello, everyone. So thank you for having me here. As George mentioned, this is based on a paper that we published this summer that was in the journal Neurology. This was a paper called Huntington Disease Reduced Penetrance Alleles Occur at High Frequency in the Population. And it attracted as well an editorial commentary with this very provocative subtitle, More Common Than You Think. So I'm going to break down a little bit in more careful terms what this means and what the implications are, because certainly that would be a pretty novel and groundbreaking finding. I'm going to step back a little bit actually and just talk about basic HD genetics. I know this is an audience that is very diverse and may not know all of the bits and pieces that go into HD genetics. So I'm going to step back a little bit and go back to some basics. So there was a study of Huntington's disease before DNA testing. Most of you know about routine genetic testing. However, before we knew what the mutation was, this is basically what the genetics looked like. So you would see someone in a family. This is a pedigree. The round one is the mother, for example, that had HD, and 2 children who also have HD. Now we know that the heritable stuff of genetics and what the mutation is embedded in, if you want to think of it that way, is DNA. So DNA is the heritable stuff of genetics. It's what we pass down between different generations within a family. And a lot of people ask, well, what does DNA look like? It actually kind of just looks like this goopy, cloudy bit on the left if you pull it out of cells and take a look. So for anyone who doesn't have the basics of molecular biology, just a quick rundown. So DNA is, you can think of DNA as the instruction manual of your cells. It tells your cells what to do. there's an intermediate called RNA that is thought of as the message of this instruction. And then the message is translated into protein, which is what actually does things in your cells. So the DNA itself doesn't exactly do anything, but it's like an instruction manual for what your cells end up actually doing. So DNA is packaged into pairs of chromosomes, as I'm sure many of you know. We're going to be looking at chromosome 4. That's the chromosome that the Huntington disease gene is located on. Now, because you have pairs of chromosomes, 1 through 22 at least, the numbered ones, that means you have 2 copies of every gene on a given chromosome. So for a gene that's on chromosome 4, you have 2 copies of a gene on chromosome 4. So as I mentioned, everyone has 2 copies of a gene. And so that means that everyone has 2 copies of the gene that's altered in Huntington's disease. So in an unaffected person, you would have 2 copies of this gene, and they would produce a normal protein that would do what it's supposed to do. In Huntington's, there is what we call a gain-of-function mutation in one of these copies, and it leads to an altered protein function, which then leads to what we know as Huntington's disease. So what does a gene look like? You can go through the basic rundown of, oh, this is the gene that is involved in such and such disease. This is the gene involved in Huntington's disease. Well, to us geneticists, a gene looks like this. And this is meant to be a little bit of a laugh slide because, the genes are very large. It's a whole strings of A's, C's, G's, T's, nucleotides that people think of when they think of genetics, but the real meat of genetics is picking apart the meaning of all of these letters. So in Huntington's, the gene that is involved is called HTT, or Huntington, and it contains what is called a polymorphic sequence. So polymorphic meaning many shapes. It comes in different varieties. And this polymorphic sequence is a repetitive CAG repeat. Actually a small chunk of the Huntington gene. And so in an unaffected person, here for example, there's 17 CAG repeats. In someone who has Huntington's disease, one of the 2 copies of the gene has more than 35 CAG repeats. So here, for example, would be 40 repeats. So as many of you know, I think, the CAG repeat occurs in a range of sizes. So most people in the population have what would be termed a normal allele or an unaffected allele, so that's less than 27 CAG repeats. Then you have a category called the intermediate allele range, 27 to 35. And then you have any allele of 36 or greater has potential to cause HD. However, the range 36 to 39 is called reduced penetrance because it may or may not lead to manifestation of the disease. So remember the pedigree I showed you before about HD genetics before DNA testing. All we knew was, well, one of the parents is affected. 2 children end up having Huntington's as well. Well, this is what it looks like after DNA testing with what we know about the CAG repeat. So in this case, the square is the father, the circle is the mother. The father has 2 repeats in the normal range, so 17 and 22. And the mother has 1 unaffected normal allele, 18, and a Huntington disease allele, which has 41 repeats. In the children, so this is just an example, a hypothetical. The son who inherited HD inherited that 41 from his mother, and then inherited a 17 from the father. The unaffected son inherited a 17 from the father, an 18 from the mother. The daughter with Huntington's disease inherited the 41 from the mother and the 22 from the father. And likewise with the daughter over here, she inherited the 22 from the father, but the normal 18 from the mother instead of the Huntington disease allele. So now we consider CAG repeats of 36 and greater as diagnostic of Huntington's. So as many of you know, you would normally come into a clinic because you have symptoms related to Huntington's. or a relative who has Huntington's. And we would then do a genetic test, and someone who has 36 or greater would be considered gene positive, and then someone who has the actual symptoms of Huntington's plus that positive genetic test would then be considered symptomatic for Huntington's. So an extension of this is this question whether all people with 36 and greater develop Huntington's disease. So it was originally discovered that people who have Huntington's have repeats of 36 or greater, but there's not quite the same question as to whether all people with those repeats develop Huntington's. So this was examined a number of years ago, where we looked at individuals who had CAG repeats between 30 and 40, and then looked at how old they were, and if they had HD. And the answer to this question as to whether all people with 36 and greater develop HD is no. So there's a range of people, so highlighted here. This is where this category reduced penetrance comes from, in fact, where some people, represented by the crosses, developed HD as early as 40 years old. However, other people, represented by the triangles, were 80 years or older and did not have signs of HD. So this is where this category reduced penetrance comes from. In other words, you have a possibility of developing Huntington's if you have 36 to 39. However, clearly not all people do. This is believed to be a result of the what we call the age-dependent onset of HD. So I think many of you know as well that the longer your CAG repeat size, generally the earlier the average age of onset of Huntington's. So on the bottom here you have the repeat size, and then on the left, on the y-axis, you have the average age of onset. So you can see in this reduced penetrance range that people tend to have age of onset greater than 60 years old. And so one sort of theory in the field is that maybe some people who have 36 to 39 would develop HD if they simply lived long enough. People do die of other causes. I mean, we develop cancer, heart disease, a variety of other things. And so what would happen if someone who had, say, a 38 lived to 90 years old, 100 years old? We don't really know the answer to that question. So to sort of get at some of these concepts of how many people actually develop HD with 36 to 39, we were lacking really a critical piece of information, which was how many people in the general population, just people off of the street, actually have these repeats. So this was the subject of the study that I'm going to talk about. We didn't really know. No one had really looked in enough control individuals, unaffected individuals, to know. I was involved in a pilot study that started this project, where we looked at repeats in British Columbia's general population here in Canada. So this is what a typical distribution of repeat sizes, what we call alleles, in the general population would look like, where 17 is the most common one. But as you look at more and more individuals, you start to see more of these less common alleles. So out of about 1,600 people from Canada, we saw 6 reduced penetrance alleles. So we decided to do genotyping in more people, in more populations, because although this was really tantalizing data, it didn't really give us, statistically speaking, a clear idea of how frequent the repeats were. We really needed to genotype more. And also we wanted to know how many full penetrant alleles there were. We didn't see any in the Canadian individuals that we looked at. So we looked at 2 more from the United States and from Scotland. The distribution in the United States sampling, this was almost 4,000 people, was quite similar to what we saw in British Columbia. We saw a similar number of reduced penetrance alleles. We also saw 2 full penetrant alleles, and these were, to the best of our knowledge, healthy, unaffected individuals. We were actually barred from seeing any clinical data by the ethics in this study. They were strictly anonymized. But clearly, when you look at individuals who are recruited as normal controls, you do see Huntington's disease alleles, both in the reduced penetrance range and in the full penetrance range. This was also true in Scotland. In Scotland, we saw a similar number of reduced penetrance alleles and also a full penetrance allele from about 1,800 people in Scotland. So it should strike you that these distributions are very similar in these three populations. And so in order to get a clearer picture of how many of these Huntington disease associated alleles, CG repeats there are, we lumped all three of them together. So in total, we saw 15 reduced penetrance alleles, so between 36 and 39, and we saw 3 full penetrance alleles. So when you think of the total number in the population in terms that people would normally think about, it means that really quite a large number of people have an HD allele that could result in HD, that is associated with HD. So in terms of just the alleles, it's about 1 in 800 alleles. And then in terms of people, when we put all three of those populations together, it means about 1 in 400 people has a repeat of 36 or greater. Now, the reason that this was a striking conclusion was not just the number, but that most of these alleles were 36 to 39. So the vast majority of people who have Huntington's disease, as I'll show in a moment, have 40 or more, what is considered fully penetrant. However, in our case, about 80% of the alleles that we saw in the general population were 36 to 39. So let's talk a little bit about how the genetics here that we've seen compares to how many people we see with Huntington's in the actual population. So this was a genotypic study. You would normally you would not use the word prevalence in this case because this is the frequency of an allele, and prevalence refers to how many people actually have a given disease or even just an appearance like blue eye color. How many people have what we call a phenotype is the prevalence of something. So how do these numbers, the 1 in 400 and then 1 in 490 of reduced penetrance, compare to the prevalence? Well, we've looked at the prevalence of Huntington's disease in British Columbia in quite a lot of detail. And the prevalence of Huntington's disease is much lower than this genotypic frequency. So in 2012, when this was studied in quite a lot of detail by a student of Michael's named Emily Fisher, we found that about 1 in 7,400 individuals has Huntington disease, manifest Huntington disease in British Columbia. So given that we know that most of the HD alleles in the population are 36 to 39, well, how many of these patients who actually come through a clinic, have 36 to 39? And the answer is not very many. It's really only about 5% of Huntington disease patients in the clinic who have 36 to 39. And when you look at the age of these individuals, most individuals with 36 to 39 are over the age of 65. So in this plot here, these are you can think of these as the number of patients, the percent of patients in British Columbia with a given repeat length in the reduced penetrance range, and in black you can see that most of them are over the age of 65. So what we did as a final stage in this study was to look at the penetrance of 36 to 39 in individuals that are 65 years or older. So the penetrance in this case means how many people with that repeat length, with 36 to 39, actually go on to develop HD. So the way we did this was we took the total number of patients over 65 with 36 to 39, so that would be the patients in black, and we divided that by the total number of people that we would expect to have 36 to 39 based on our genotyping data. And we call this a direct penetrance estimate of HD for 36 to 39 in people who are over 65 years of age. So this is as confusing as it will get. Don't worry. Our findings were in the gray bars. So at 36, we in fact didn't see any people in British Columbia who had had HD with 36 in 2012. There have been some in the past, but there weren't any in 2012. So clearly the penetrance of that is very, very low, but greater than 0. For 37, we found that only about 1/5% of individuals with 37 appeared to have HD over the age of 65. And then at 38, it would seem that only about 2% of individuals who have a 38 actually have HD over the age of 65. Now, in the white bars were estimates of the penetrance of these alleles from the clinic. So naturally, people who come to the clinic are people who have HD. And so historically, we believe it's been overestimated how many people have Huntington disease with 36 to 39. Now, our numbers, so the 0.2% and 2%, these really should be thought of as minimums. There's many reasons why we wouldn't ascertain people who have a 36 to 39 over the age of 65. This was only looking at people we actually saw in our prevalence study in British Columbia. So why do we see these alleles so rarely in the HD clinic? Well, as I just showed, as I alluded to, the penetrance of these alleles for Huntington's disease is probably very, very low. So there are many more people with 36 to 39 than we previously thought. But the good news is that it seems like fewer of them actually develop HD than we previously thought. A secondary explanation that is quite interesting that we're exploring is that late onset HD, so HD over the age of 60, that results from 36 to 39, if you remember the curve that I showed you before, may not actually be recognized as Huntington's disease. Many times people who have 36 to 39 are the very first person in their family who have HD. They don't have a family history of HD. And so it could be that individuals who have a late onset with 36 to 39 don't ever get referred to a neurologist. They may never even go to a clinic. So we believe that it's probably a combination of these two possibilities as to why we so rarely see 36 to 39 in the clinic versus how frequent it really is in the general population. So as I said, there's some interest in this, particularly from the clinical community, from neurologists, as to whether there maybe is a spectrum of mild HD phenotypes in the elderly. And so this was something that was touched on in that, that commentary that I mentioned at the very beginning of the presentation, that the threshold for testing in elderly individuals who maybe have suggestive phenotypes of Huntington's, suggestive signs of Huntington's, should maybe be lower. Right now, there's many people who would only test for Huntington's if you actually have a family history of Huntington's. So there's many reasons based on the genetics to believe that maybe more people who are over the age of 65 who maybe have subtle symptoms should be tested. And maybe we'll have a clearer picture of how many people with the 36 to 39 repeats actually get Huntington's disease, and maybe they actually have milder symptoms than what someone would have in a full penetrance in the full penetrance range. At this point, we don't really know, which was part of why these findings are quite provocative. So it's a fairly short presentation, but I realize that there's quite a bit packed into it. So I'm just going to acknowledge my colleagues here at the Center for Molecular Medicine and Therapeutics at UBC, and then our collaborators at the University of Aberdeen and at the Coriell Institute in New Jersey. And now I'll open it up for George to take questions. I'd be happy to respond and clarify anything that I presented. So thanks very much.
Thanks, Chris. That was really great and very clear. So folks, if you do have any questions, feel free to, like I said, type them in at any time, and we'll ask them of Chris. One here is, it's my understanding that if one of your parents has HD, then the children have a 50% chance of having HD. In my family, it's closer to 80%. I was wondering if the percentage is proportional to the number of CAG repeats.
So the chance of inheriting?
Yes,
So the
I
chance
guess.
so the chance of inheriting would be 50-50. So that's based on just a 50% likelihood of getting the normal allele from a parent who has Huntington's versus the HD allele. However, that doesn't change based on the number of repeats. The number of repeats does not affect the chance that you will inherit it. It will always be 50%.
Great. There's another, Chris. If one does have the CAG from the affected parent, is the number going to be the same number as the parent?
No, frequently it will change. It is often the same. However, it can go up and it can go down, particularly with regards to the expanded CAG repeat.
Right. Another question is, how many of the patients with intermediate values were male versus female? The person asking the question says this may have an impact on the next generation who has a higher repeat length.
Mm-hmm. So that's interesting to ask. I did look at that because in one of our cohorts from the United States, we did have some basic demographic information about race, gender, and in terms of intermediate alleles, it's as you would expect. So it's just 50-50. So among people who have an intermediate allele, half of them are male and half of them are female. So there's there seems to be no bias by sex for having an intermediate allele.
Great. So this is a question that I knew would come. It's asked a lot of HTSA and of other folks in the community. Is the commonly used statistic of 1 in 10,000 people being affected or symptomatic for HD. Based on your data, do you believe that that is a low number?
Yes. So as George alluded to, this is a provocative question. I personally think that it is higher than 1 in 10,000. I would caution to say it's not much higher than 1 in 10,000. And part of it depends on what ends up getting classified as Huntington disease. So most of the people who are listening to this webinar, I think, are from families where there are full penetrant alleles. In that case, I would say it's 1 in 10,000 or slightly higher. However, there may be people, as I mentioned in the presentation, who don't have full HD, and if you put those into the prevalence number, that would increase it as well. But 1 in 10,000 is a good ballpark number. It is not dramatically different than 1 in 10,000. The real mystery is why are there so many of these alleles and so few of them with 36 to 39 actually get Huntington disease.
Great, thanks, Chris. Another question. If an undiagnosed male dies with a CAG in the range of 36 to 39, could they pass the HD gene to a child who would have a greater CAG length?
So the answer is yes, absolutely. And historically, we had thought of new mutations for Huntington disease as coming from intermediate alleles. So the repeats that I'd shown from 27 to 35. However, one other conclusion of our study, which is also very interesting and provocative and important, is that actually many new mutations for Huntington's disease may be coming from people who have reduced penetrance alleles. So yes, the answer to your question is yes. There may be, for example, a father who has 36 and never develops HD. However, they may have a child who has 42. And so really, when you're thinking about where new HD alleles mutations come from, people who have these 36 to 39 alleles should now be thought of as an important source for new mutations, yes.
Thank you. Question was asking you to revisit the discussion of prevalence and penetrance for folks with 39 repeats.
Mm-hmm.
I'm not, but I know that you didn't find anyone in your 77 plus thousand folks with 39 CAG.
Right. So let me just go back briefly to this this penetrance estimate figure. So yes, there's no data for 39 in this figure, and the reason there's no data for 39 in this figure is, like George alluded to, we did not see anyone in the general population. So that would mean from British Columbia, Scotland, and the USA with a 39. Now, it should also be apparent to you that among people who have reduced penetrance alleles, so 36 to 39, 39 is actually the most common. So if it's the most common in the clinic, but it's less common in terms of genotypes as 38, you would expect the penetrance to be higher than this number that I have on the right for 38. However, we could not directly calculate it. So right now, the answer to your question is unfortunately a bit unsatisfying. We don't have the data to make an estimate for 39, but it's higher than what we would estimate for 38.
Great, thank you. Let's see here. My CAG repeat is 26 and 28. I have a son that is thinking of having children. Can his CAG repeat? Let's see what the question is. Can his CAG repeat, I'm guessing, is expand to a larger repeat?
So the answer is yes. However, it's highly dependent on how long his repeat is. So if, for example, he inherited the 27, and the 27 is exactly the same as it is in you, the risk is very, very, very low. So we had done a study just a couple years ago on how frequently intermediate alleles expand based on their size. And the risk is much higher for people who have, say, 33, 34, 35, quite low for people who have 27, 28, 29. But that would be something you should probably discuss more with a genetic counselor as well.
Great. We've got a ton of questions, Chris, which is great. This is probably the most questions we've ever had on a research webinar, which is really
good. It's a popular topic.
It is a popular. I knew it would be. So do you think that this penetrance estimate can be applied to people with 36 to 39 repeats who have others with symptomatic HD, or do you think other common genetic backgrounds may make penetrance in those families higher?
Mm-hmm. So that's an excellent question. The answer as to whether those penetrance estimates apply to people and families is no. So in terms of penetrance estimates for people who have relatives with Huntington's disease, I mean, this is evolving data, this is evolving knowledge, but I think a neurologist or a genetic counselor would be inclined to use the numbers here in white. Now, the reason for that, if not everyone in the audience understands what was asked, is that there's other things in your genome, there's other things in your environment that may result in the onset of HD. Not many of those factors have been characterized, really only a few. And so at this point in time, if you had a reduced penetrance allele, 36 to 39, and you had a relative who had Huntington's, I mean, I would be inclined to say that there's a higher risk than the numbers that we published here on the right in gray. Because strictly speaking, that would be the penetrance for someone completely from the general population, not associated with an HD family. But how much that risk of penetrance varies from, say, someone from an HD family versus someone from the general population, we don't really know. We don't really know how big that gap is.
Yep. Thanks, Chris. Let's see here. Try to get through some of these. Was the population with increased prevalence in Scotland included in this analysis?
There were no clinical samples from Scotland included in this set. These samples were from the general population of Scotland. So yes, it is believed that there is an elevated prevalence of Huntington disease in Scotland. However, statistically speaking, we didn't see enough Huntington disease-related alleles to say whether that number was actually higher in our Scottish sample. So it could be that if you looked at many more controls from Scotland, we looked at about 1,800 people. If we looked at 5,000 people from Scotland, it could be that you would see actually a higher number of HD alleles. However, we didn't look at enough to say whether that was true or not.
Great. Let's see. Sorry, there's a lot of questions. Trying to filter through some of are asked multiple times. Are there any studies, have there been any studies regarding, and I think we kind of touched on this, regarding gender and the increase in repeats between generations?
So most new expansions, most new mutations for Huntington's disease come from a father. So Huntington disease has what's primarily is called paternal, there's primarily paternal inheritance of a new mutation. So there is a predisposition, particularly in the intermediate allele range, so 27 to 35. There's a predisposition for expansion when that allele comes from a father. This is not as pronounced in the Huntington disease range. There's still a bias towards expansion from fathers. However, there is quite a lot of instability when the allele comes from a mother as well. Principally, the bias is in the intermediate allele range.
Great, thank you. Another question about, you raised an interesting point towards the end of your presentation about the elderly. As we live longer, we may see more and more people with symptoms of HD. And this question is, should we start testing all elderly patients with dementia or abnormal movements for HD?
So a very, a contentious train of thought resulting from this study is what sort of data are we going to have in the future on the CAG repeat from people just in the general population? I think a lot of you know that we're moving into an era of personalized genomics. More and more people are having their genomes sequenced. In the future, we're likely to have a lot more information about our genome rather than fewer. And so my personal thought, I'm kind of going off the script a little bit for a moment, is that I really think we're going to come at this in a different direction. It's not going to be that we're going to size people who are elderly, who maybe have suggestive phenotypes or symptoms. I think it's actually more likely that we're going to size everyone in a given population and end up finding some people who have associations with those repeats. However, it drifts into some very complicated issues as to whether services that would provide genome sequencing would do this. The Huntington disease repeat, of course, is associated with Huntington's, and maybe it would be omitted. But my personal thought is that we will end up finding out answers to some of these questions as many more people find out incidentally that they have these repeats. But that's really my personal opinion. It's not representing any organization. So.
Thanks, Chris. So back to the 1 in 10,000. Another question related to the 1 in 10,000 number that's always thrown around for HD prevalence. Your data that you showed, if I remember correctly, was 3 people with full penetrance allele in 7,315 patients or individuals.
That's right.
Which is roughly about 1 in 2,500 or so. How does that contradicts the 1 in 10,000 number fourfold? How can you explain that?
Well, an interesting aspect of the study was, in fact, that was exactly the number that we expected given the 1 in 10,000. So 1 in 10,000 actually means how many people who have HD. They are actually symptomatic. So 1 in 10,000 does not mean how many people actually have a full penetrant allele. Because as you know, you develop HD in midlife or later. So the frequency that you mentioned, so 1 in 2,500, that includes, say, someone who's 20 years old who has a 42, 30 years old who has a 41. Those people don't have HD yet, most likely. However, they're actually included in the genotypic number. So it was actually it was an interesting conclusion of the study that the 1 in 2,500, based on some prior work by geneticists many years ago, was in fact how many we expected based on the 1 in 10,000 that have HD symptomatically.
That's great. Great answer. Thank you for clarifying that, Chris. Let's see. All right, we're getting there. And we have a few minutes, Chris. Let's see. We've got. Will each generation have an earlier onset as you go further and further down?
Generally, yes, but not necessarily. So the expanded repeat is unstable in general. It does have a tendency to expand, but because it is unstable in general, it also has the potential to contract. So it is possible for someone who inherits an allele to have an earlier onset than their parent. However, generally speaking, when you look at trends, it tends to become longer, and the onset tends to be earlier as you go down through generations with the repeat.
Another question or statement and question is that for prevalence of genetic abnormalities, this study is relatively small, I guess, in terms of sample number. Is there a larger study being planned? I don't know if it was in your manuscript or the commentary that accompanied it. There was a reference to maybe a genome screening of 100,000 people, a study that's going on in Europe. That would something like this be included, a study like that, or is there something else being planned to get even higher numbers?
Right. So the answer to that question is basically no. There is no such study planned at the present moment in time. And to be clear why, it's that the technologies that are being used in some of these larger studies, like the 100,000 individuals, those technologies cannot size the CAG repeat. They are unable to report the length of the CAG repeat, and the reasons are rather technical, but we were able to do this study because we developed the predictive test for Huntington disease. In terms of us, we would like to do this in larger numbers. We would like to do this in groups of people who have clinical data. However, in terms of the study that was mentioned in the editorial, so the 100,000, no, the CAG repeat is not included in that study or similar studies.
Great. And there's some questions about, in studies such as Enroll, would it be helpful for everyone who has been tested for HD and knows their CAG repeat length to include that in the study? I mean, I think that is included in the Enroll. As part of the study, you're not going to know, but the CAG length, you are genotyped everyone who's in the Enroll study, so they will have that information.
Well, it gets back to this question of whether the rates of Huntington's you see among people with 36 to 39 are reflected by people who come from HD families and come to the clinic currently. So the novelty of this study was that we looked at people who didn't have any association with HD whatsoever. And so I'm not sure actually that looking at Enroll would add that much more versus what we currently know.
This is a little, so I'll try to answer this. interesting one. This was a question came from about a year back. We had a speaker, and I think it was Francesca Cicchetti, who also from Canada, who had really interesting data about the transfer of the Huntington protein between cells. And I think to answer this person's question, they said that that speaker said there's a possibility of acquiring the disease through a blood transfusion because the blood cells might contain the HD protein or cell. Do you have any info on this? So I think what you're referring to is that there are data that the Huntington protein could potentially go between cells, but it's possible. So if somebody with Huntington's disease donates blood, that Huntington protein could get into the unaffected person, but it's not going to cause, I don't see how a blood transfusion is going to cause Huntington's disease. They're not transferring the gene. I don't know if you'd agree with that, Chris.
No, I definitely would agree with that because Huntington's develops over decades of someone's life, and what a lot of people don't realize is that the mutant protein, the protein that results from the expanded CAG repeat, is present from birth in an individual who has an expanded repeat. So having some protein from a blood transfusion, I think, would be far from sufficient to result in any sort of HD, any sort of Huntington disease,
And also for the person asking the question, that protein that is around, that doesn't stay around forever. Proteins are chopped up and gobbled up by the body and recycled. So that even if there was some Huntington protein in the body, it would not stick around for a lifetime to manifest HD in that unaffected person.
Exactly. Yes.
So let's see. Here's a long one. Let me see how we can get through this one. I think this might be our last one. My grandfather died at 87 and not diagnosed with HD. He did pass it to my mom, who had 38 repeats and died at 64. My brother had 38 CAGs and died at 57. Do you look for people who had a range of repeats who have died? Sure I follow that one. My other brother was 35 and told he was in the gray area. He may or may not get it, which is going to be that 36 to 39 range that we've been discussing today. He's been seeing neurologists every year. He's 51 and was told he didn't need to come back because he didn't have HD. Would you agree? I don't know if you can answer that one, Chris. That's it.
Well, so it's important to note that these categories are assigned based on clinical data. So when we say that someone could have Huntington's with 36 to 39, it means that we have never seen someone with Huntington's with 35. So in the case of your brother, who has 35, I would say that I would probably concur with the definition, with the recommendation of the neurologist, because we have never seen someone who has CAG35 who has Huntington's disease.
Just another question that's totally not necessarily related too much with your talk, but can HD patients donate organs? I believe they can. I don't know if you know the answer to that.
I would think so. I can't imagine there's a prohibition on that.
Yeah, that's a great question, and I'm going to look into that. But I'm pretty sure that
I don't know the answer. I wouldn't think that would be a problem, but yeah, I don't know the answer.
Great. So I guess the last one here is just if you had any advice for those of us with reduced penetrance with respect to living our lives.
I wish I knew the answer to that. Unfortunately, we're not going to know the answer to what can be done to change lifestyle to reduce the risk of HD until we have more data. I mean, until then, there's certainly recommendations based on people who have full penetrance alleles that are worth following. Stay active.
Exactly. I think, it brings me back to our convention. Robert Pacifici, who's the CSO of CHDI, spoke, and I think he was the one who said, we're all gene positive for something or at risk for something.
Right.
The best advice, whether it's you're at risk for reduced penetrance for HD, or maybe it's Alzheimer's or cancer we just don't know, is to take care of your body, eat, sleep, don't drink, don't smoke. I think, and stay active. It's kind of the best advice for everything. It's kind of a cop-out, but it's true.
It is, yeah.
But anyway, well, I think hopefully we've asked most of these questions. I apologize if I left any of them out. I'm trying to go through them as quickly as possible. We are running out of time. But I do want to thank you, Chris, for a really great presentation. This was really clear, and it's going to be hopefully a great resource for the community as these questions come up. So thank you again, and good luck with finishing up your PhD.
Thank you. Yeah, it was a great pleasure. Thank you all for attending and having me here.
Take care, and we'll see you again on the next HGSA research webinar. Bye-bye.
Okay. Bye-bye.